Hello there, welcome to ShineTech's YouTube channel. In this video I'm going to talk about cell injury, cell death and causes of cell injury. So causes of cell injury are hypoxia, toxins, infectious agents, immunological reactions, genetic abnormalities, nutritional imbalance, physiological agents and aging. So these are the causes of cell injury. But now let's look at these causes of cell injury in detail. What is hypoxia, toxins, what is all about infectious agents, and all of these factors which causes cell injury. So hypoxia refers to deficiency of oxygen. Okay, so we have a reduced oxygen supply to a certain tissue, and that is what we refer to as hypoxia. It is it is the most common cause of cell injury. So the most common cause of cell injury is hypoxia. Okay, and mostly hypoxia is caused by ischemia. So ischemia, here we are talking about reduced blood supply to a tissue is ischemia. We have a blood vessel which has been occluded and blood supply to a certain tissue is reduced. That is ischemia, and ischemia is the most cause of hypoxia. So hypoxia is of several types. There are 4 types of hypoxia, which are, we have anemic hypoxia. So anemic hypoxia is due to decrease in the function of hemoglobin. Decrease in the functional hemoglobin. So the functional hemoglobin is decreased, or the number of red blood cells having functional hemoglobin is reduced. That is anemic hypoxia. And the capacity of blood to carry oxygen is reduced, and inadequate levels of oxygen around the body. Okay, so we have inadequate levels of oxygen around the body. Why? Because the capacity of blood to carry oxygen is reduced due to decrease in the functional hemoglobin. Then another type of hypoxia is hypoxic hypoxia. So hypoxia is due to inadequate saturation of blood oxygen as a result of reduced supply of oxygen in the air, decreased lung ventilation, or respiratory diseases. So here we have reduced oxygen in the air. Okay, this is observed in high altitudes where oxygen levels are very low, and that can cause hypoxic hypoxia, or decrease in lung ventilation. Okay, ventilation of the lungs is decreased, that is hypoxic hypoxia. Or respiratory diseases such as asthma can also cause hypoxic hypoxia. So apart from hypoxic hypoxia, we have stagnant hypoxia. Stagnant hypoxia. So as the name says, this arises from a decrease in blood flow. So the flow of blood to the tissue or an organ is reduced, preventing adequate blood supply to the tissues. Okay, we're going to have low blood supply due to decreased blood flow. This is observed in heart failure, heart attacks. Okay, so the organs will receive less blood supply, and if less blood supply is received, meaning even the oxygen levels which are supplied are going to be reduced, and that will cause, is referred to as stagnant hypoxia. So the last one here we have histotoxic hypoxia. So histotoxic hypoxia, here we're talking about oxygen is delivered to the tissues, but they fail to utilize it effectively because the cells are damaged and cannot extract and absorb oxygen from the circulating blood. Okay, so oxygen here is being supplied, but the cells themselves, they are damaged and cannot extract or absorb that oxygen from the circulating blood. That type of hypoxia is histotoxic hypoxia. So hypoxia can cause cell injury, and it is of 4 types. As I've already explained, we have histotoxic hypoxia, we have stagnant hypoxia, and then we have hypoxic hypoxia, and we have anemic hypoxia. So apart from hypoxia, another cause or factor which can cause cell injury are toxins. So toxins can cause cell injury. So toxins, we're talking about air pollutants, insecticides, carbon monoxide, asbestos, cigarette smoke, ethanol, and drugs. So these are toxins which can cause cell injury. So apart from toxins, we have infectious agents. So infectious agents here, we're talking about all types of disease-causing pathogens. All types of disease-causing pathogens, which are viruses, bacteria, fungi, and protozoans. So these pathogens can cause cell injury. Then apart from infectious agents, we have other factors, we have other causes, which are immunological reactions, genetic abnormalities, nutritional imbalance, physiological agents, and aging. Okay, so immunological reactions here, we're talking about autoimmune reactions, allergic reactions. Those can cause cell injury. And genetic abnormalities, we are talking about deformed DNA. And you know that if the DNA is deformed, what will happen is that even the proteins which are going to be translated from there, they are going to be deformed as well. We are going to have proteins which are misfolded, accumulating inside the cell. So when that happens, the cell will be injured. Nutritional imbalance. Here we are talking about feeding habits, where someone eats an imbalanced diet, which can lead to obesity, and obese can cause diabetes mellitus type 2, which is simply maybe the insulin is not working well, or the cells are resistant to insulin, and that can lead to cell injury. Okay, imbalance, nutritional imbalance. Then physical agents. Physical agents, we're talking about the increase in temperature, we're talking about trauma. So all those are physical agents. Atmospheric pressure is also a physical agent. Then aging can also cause cell injury. Okay, so we're done with causes of cell injury. Let's move on now to reversible and irreversible cell injury. So in my previous video, we talked about adaptation. Okay, all these types of adaptation, I explained them. Now here we're going to talk about reversible and irreversible cell injury. But first of all, we have a normal cell here. Then this normal cell, there is a stress. This is being stressed. So this stress is going to cause adaptation. Okay, so adaptation. So the cell, if the cell is stressed, is going to adapt to that situation. So if it fails to adapt, inability to adapt, then it is going to be injured, and that injury is called reversible injury. This injury, if resolved, then the cell will go back to its normal function. But if it is severe, then the cell will progress to irreversible injury. Then irreversible injury, now here the cell will eventually die, and it can die in 2 ways. It can die via necrosis or apoptosis. So let's look at reversible cell injury. So reversible cell injury, by definition, is simply the stage of cell injury at which the deranged function and the morphology of the injured cell can return to normal if the damaging stimulus is removed. So here the cell is injured, and if that damaging stimulus is removed and that cell returns to its normal function, then that kind of injury is what we call reversible cell injury. How does it happen? Or what are some of the activities which takes place under reversible cell injury? So here, in the first place, I mentioned that ischemia is also one of the cause. Hypoxia is one of the cause of cell injury, and hypoxia is caused by ischemia, obstruction of arteries. So blood supply to the tissue is reduced, hence hypoxia. So hypoxia, we know that mitochondria is sensitive to oxygen. Once the oxygen levels are reduced, mitochondria will be dysfunctioned. So we're going to have mitochondrial dysfunction. Why? Because of hypoxia. Then, once the mitochondria is not working well, we are going to have reduction in oxidative phosphorylation, which is going to cause reduction in ATP. ATP won't be there. Why? Because mitochondria is not working well. So here, mitochondrial dysfunction is the first change in cell injury. So reduced ATP or no ATP production will cause the sodium-potassium pump, which is found in all cells, to stop working. So once this pump is not working, we'll see consequences, what will happen. But before that, this pump, sodium-potassium pump, so this pump, what it does, it is on all cells. Okay, so what it does, it removes 3 sodium ions for every 2 potassium ions. If it is not working, then sodium will start accumulating, because this channel is the one which removes sodium. So we're going to see accumulation of sodium inside that cell. So we know that where sodium accumulates, water follows. So here we're going also to have influx of water. So influx of water is going to cause cellular swelling. So cellular swelling is the first morphologic change in reversible cell injury. So the cell is going to swell, organelles inside the cell are going to swell as well. Okay, so organelles inside the cell are going to swell as well. So the cell is going to swell, and also we are going to observe loss of microvilli. Okay, microvilli, we are going to see the loss of microvilli. Why? Because the cell is swelling. So microvilli on the cell, these are the microvilli. So when the cell swells, what will happen is that it is going to lose, it is going to be like this. So the microvilli are going to be lost. So this structure here is going to be lost because of cell swelling. And another thing which will be observed here are what we call membrane blebs. So membrane blebs, okay, I'm going to explain this in the next slide. Okay, so membrane blebs are also going to be formed in reversible cell injury. Okay, so apart from this, we are going to see anaerobic glycolysis. Why? Because ATP is no longer being produced. So the cell will undergo what we call anaerobic glycolysis. So anaerobic glycolysis is going to produce lactic acid. Then lactic acid now is going to lead to reduced pH. So the inside the cell will be acidic. Okay, so we are going to have acidosis there inside the cell. Then apart from that, we are also going to have depletion of glycogen. Glycogen is going to be depleted. But first of all, let's see the consequences of reduced pH. So reduced pH is going to cause what we call clumping of nuclear chromatin. So nuclear chromatin is going to clump, it's going to shrink, it's going to become small in size. So this is a first nuclear morphologic change under reversible cell injury. So the nuclear morphologic change is clumping of nuclear chromatin. Okay, so apart from clumping of nuclear chromatin, we are going also to observe detachment of ribosomes from the rough endoplasmic reticulum. So we know that these ribosomes are attached to the rough endoplasmic reticulum, but when the water accumulates inside the cell, the rough endoplasmic reticulum is going to swell. So as it swells, some of the ribosomes are going to be detached. So once they are detached, we know the function of ribosomes is to produce proteins. The synthesis of proteins takes place in by the help of ribosomes. So if they are detached from the rough endoplasmic reticulum, we are going to observe a reduction in protein synthesis. Okay, so this is what happens under reversible cell injury. So the membrane blebs, which I mentioned earlier, so these are simply protrusions. So on this diagram here, this is a normal cell, then it was stressed and it became reversibly injured. It became injured. So once it was injured, we saw there was the accumulation of water. Then that accumulation of water caused the formation of membrane blebs. So membrane blebs, these are simply pouches or protrusions which are formed by the membrane to accommodate water. So since water is accumulating inside the cell, we are going to see pouches, these pouches there, which are formed to accommodate more water, and those pouches are what we call membrane blebs. Then apart from membrane blebs, under reversible cell injury, we are also going to observe myelin figures. So myelin figures are simply membranes which are membranous walls which are detached from organelles, membranes of the organelles inside there, okay, and are made up of phospholipids. Okay, so in a nutshell, the activities which takes place under reversible cell injury are: the first one is cellular swelling, which is the first morphologic change in reversible cell injury. But the first change is mitochondrial dysfunction. The first morphologic change is the cellular swelling. Then from there we have swelling of the mitochondria and endoplasmic reticulum. And then we also have loss of microvilli. Microvilli are going to be lost, membranous blebs are going to be formed, and myelin figures are also going to be observed. And myelin figures, as I said, these are membranous walls or lamellated aggregates of phospholipids derived mainly from damaged membranes of mitochondria, endoplasmic reticulum, or the cell membrane itself. Those are myelin figures. So that's all about reversible cell injury. Now let's move on to irreversible cell injury. So under irreversible cell injury here, now we are talking about severe membrane damage. So the cell membrane is severely damaged. So once the cell membrane is severely damaged, we are going to see accumulation of calcium inside the cell from the extracellular fluid, and increased calcium influx in the cell will activate enzymes inside there. We know that calcium activates enzymes inside the cell. That's why it is kept outside the cell. So certain enzymes are going to be activated by calcium. One of these enzymes is phospholipases. And phospholipases, these are enzymes which are going to work on lipid structures, okay, such as the cell membrane itself. So the phospholipases are going to degrade the cell membrane, and the membrane of the mitochondria are going to be damaged. So once the membrane of the mitochondria is damaged, we are also going to observe accumulation of calcium inside the mitochondria, and this is what we call MFA's densities. So the densities of calcium inside the mitochondria here. Then from there, proteases also are going to be activated by the calcium. Then proteases are going to cause cytoskeleton damage. Then apart from this, we are also going to see nucleases are going to be activated by the calcium. The nucleases are going to degrade the DNA inside the nucleus. It's going to be damaged. And then from here, the cell will eventually die. So in a nutshell, we have looked at the causes of cell injury, and then we have also looked at the reversible and irreversible cell injury. Then in the next video, we are going to look at necrosis, apoptosis, and calcification. So thank you so much for watching this video. Don't forget to like, subscribe, and leave your comment in the comment section below.